Oh, welcome everybody, especially my QBRI colleagues. It's been quite a few months since I've seen most of you. So we're excited that soon we'll be getting back into the labs and starting to find some level of normalcy. So really looking forward to that. But in addition to welcoming our QBRI colleagues, I want to welcome everybody else who's come in to be part of our new webinar series. Looking for silver linings in COVID infection. I think this is one of them, which is we can get world-renowned speakers, with a little bit of preparation to pop into our place of wherever we're watching this and start hearing some excellent science. So for my QBRI colleagues, we're going to be doing this for quite some time to have a regular seminar series, and we've set up a committee to start recruiting speakers to keep us informed and keep us engaged in scientific activities around the world. So look forward to that, and we're always looking for good speakers to invite in through these virtual setups now. So if you have speakers that you want to bring over, get in touch with me or the committee that's being formed to do that. So there's a lot of people from outside QBRI who don't know me. I'm Dr. Larry Stampton. I'm the director of the Neurological Disorders Center at QBRI. We're having our kickoff seminar here, this series, and we were excited to have Dr. Swain Chen from Singapore. So I've known Swain for many years. In fact, I was involved in recruiting him to the Genome Institute of Singapore, where he's a group leader. His primary appointment is as an associate professor at the National University of Singapore School of Medicine. And just a little background on Swain. Swain did his undergraduate at Harvard, then he did an MD PhD at Stanford, and then he went on to do a fellowship with Jeffrey Gordon at Washington University in St. Louis, where he really started carving out his expertise in thinking about pathogenic bacteria. And that's really what he's been doing since he's become an independent group leader and professor in Singapore. And so we're really excited to have him here to talk about his interests. And you'll see from his talk that, for many years, we've often thought about, an infection being, one particular pathogen, and, once we get rid of that pathogen, we're fine. But now we're learning that pathogens live in colonies and big groups, and it's a subset of those within that big population that makes some genetic transformation that ends up causing pathogenesis in humans or fish or other livestock. So this is really been a golden opportunity for the type of work that Swain does, which is to use genomics and computational tools to figure out that rather than looking at one individual organism, really be thinking about this on a population basis, what some people call pangenomics. Again, this is very cutting edge stuff. I think the marriage of genomics with epidemiology and infectious disease has really broken some exciting new ground in being able to understand the spread of infectious diseases in communities. And Swain will talk about his work with the pathogenic E. coli that causes urinary tract infections, and also give a report on some exciting work he did in Singapore to track down using these types of tools and techniques for looking at group B Streptococcus that is a foodborne pathogen causing pathology in humans. So again, it's exciting to have him here. He's been pushing this forefront of using high throughput genomics, and now more and more into single cell genomics to really start figuring out what are the pathogens, how did they evolve, what are the molecular basis for how they cause pathologies in people. And so this is a very timely talk, as we're thinking about infectious diseases, vis-a-vis COVID and how it's impacting our lives and how we can control it and how we can understand it and how we can be prepared better for the next time something starts emerging, that we can get a handle on it before it becomes so far of an outbreak as we have now. So I'll soon turn it over to Dr. Swain, but let me just, a few points here. So this talk is being recorded. So if you need to run away, your kids start screaming, whatever, you can come back and listen to it. The recording will be stored somewhere, and you'll see that information later. We will have a Q&A session. We'll do that at the end. So if those of you working on this WebEx events, you'll see you have a field where you can write in questions. So please do write in some questions, and as time permits, we'll relay those on to Dr. Swain for him to address at the end of this talk. So without further ado, I'd really like to turn it over to Dr. Swain Chen from Singapore. Excited to have you on board, Swain, and looking forward to hearing your presentation.
Hey, thanks for the very warm introduction, Larry. As he mentioned, you know, I've known him for quite some years when he was back in Singapore. And so it's also a great honor for me to be giving this sort of kickoff webinar for you guys in Qatar. You know, I had a chance to visit a few years ago. There's another good friend of mine from Singapore days, Prasanna Kalakar, who's also out there. And so really, you know, it is true. Things are a very different time right now. And, you know, having this opportunity to give this seminar, which is going to talk about some of the work we've been doing in the lab over the last couple of years, looking at virulence mechanisms, I think all of that really, it's a very special time, and it actually sort of ties together a lot of the things that we've been working on for many years. And so I think, you know, hopefully you'll find some of the connections, I think, at a deeper level. Although we're studying different pathogens in COVID, there are some deep connections, I think, in terms of what are the important questions for infectious disease that we've been really trying to address. And hopefully I'll be able to convince you that, you know, there's still a lot of work we need to do, not just on individual diseases, but on our entire process of how we understand infection and how we're using genomics to do that. So let's see here. You know, I don't really need to explain this slide. Okay, so this is why I'm giving a webinar. This is why you guys are holding a webinar. This is why our labs, many of the labs across the world, are shut down. So this was just, you know, the status. This is from Johns Hopkins University. They have a pretty good visualization of the situation of COVID. And what I want to do is say, you know, really, why is this so concerning? And what are the things that are stopping us from solving this problem? And, you know, here's an example of one of these, right? And so again, I think I probably don't need to say too much about this. There's a lot of unknowns, right? So there was a lot of back and forth about whether chloroquine or hydroxychloroquine was a good treatment or a possible treatment, right? And then actually the timeline is very compressed. So if you take a look here, 16 March and then to 27 March, you know, this question right now, we just don't know what to do, but we need to have choice. We need to make choices when you have sick patients. And eventually, you know, the hope is that in relatively short order, and this was quite fast for chloroquine, that, you know, we either figure out whether it does work or it doesn't. Okay, but if you think about this situation, right, it really comes down to virulence mechanisms, which is what my lab and what my research work has been trying to understand for many, many years. And what do I mean by virulence mechanisms? Well, in the case of COVID-19 or SARS-CoV-2, you could kind of say, like, you could kind of ask a few questions that are all related to virulence mechanisms. And these are all relevant questions. And I think, you know, people now start to really understand why it is that these things are so important, right? SARS-CoV-2 is a very similar virus. If you just look at DNA sequence or just look at the nucleic acid sequence, it's very similar to the original SARS from 2003, but it seems to behave very differently. Why is that? We think it's probably encoded in the genetics, but it's very hard to figure out what that is. Just on the last slide, I showed you those things about chloroquine. How do we treat COVID-19? That's a big question. And, you know, I think many of us doing research would say, well, if we understood how exactly this virus is causing disease, maybe that would help us to develop new treatments or to understand how we could prevent it. And then there's another question where if we really truly understood how it is that bacteria or viruses or just any kind of pathogen causes a disease, could we actually try to anticipate this? Because I think one of the clear things that has been going on is the world essentially was unprepared for COVID-19. There may have been lots of warnings. There may have been people that sort of said, we should be watching out for viral pandemics or contagious, respiratory diseases. But at the end of the day, part of the reason this has been so big is that we were essentially unprepared for this as a global community. Okay, so then I'm just going to touch one more on this, right, which now relates this to genomics, and it relates the virulence mechanisms, or this first question here, where how is SARS-CoV-2 different from the original SARS back in 2003? Again, we can go back to the genetics, and people are looking at these sequences. It's been an exciting time over the last 20 years for genomics. We can get a lot of nucleic acid sequences, and those can be used, right? And so you would see these articles. This was not so long ago. Scientists say a now dominant strain of the coronavirus could be more contagious than the original. And this article was, when they say now dominant strain, that was looking at the nucleic acid. It was looking at the RNA sequence and saying, hey, there's some differences here. And those differences seem to correlate, or we see, one sequence difference that seems to be, more present in the Americas than in Asia or China, or where the virus started. And maybe that means it's more virulent, or it got more dangerous, or is causing more severe disease. Okay, but I think, there has been a lot of work. There have been a lot of people focusing attention on this, and the sort of more say reasoned kind of consideration of those sequence differences. At the end of the day, it's just like the chloroquine story. There's an intermediate phase where essentially we just don't know. Okay, and so this was this guy Ed Yong. He writes for The Atlantic, and he wrote this article about the problem with stories about dangerous coronavirus mutations, which is sort of a direct commentary on these types of articles that were essentially just looking at a few sequences and saying, we now know how it works. It's exactly the same kind of thing as saying, oh, chloroquine worked, in a very small study, and so now we should sort of maybe give this to all the patients across the world. But then it ends up being that normally, or chloroquine ends up on balance harming patients instead of curing them, right? So there's a phase where we don't know, and really the big question is how do we get past that? Okay, and so these are these questions about virulence mechanisms. They're highly relevant today. You see them underlying the key debates, the key unknowns, the key news articles that you're seeing in the newspapers every day, right? And if I could kind of generalize this away from COVID-19, or generalize this away from SARS-CoV-2, virulence mechanisms essentially deal with questions. Those exact same questions would, in a more general sense, would be how does any pathogen cause disease? Specifically, how does it cause disease? And then if we knew how it causes disease, does that help us to develop treatments or to prevent the disease from occurring? And then if we actually truly understood this on a wide scale, if we really understood how pathogens interact with humans, could this actually help us to predict? Could this help us to foresee when new infections are going to arise or evolve? And that's, of course, like a big long-term goal for the lab. So, Larry mentioned that there's a few bacteria. So I work on bacteria and not viruses. Of course, like everybody else, we've all learned very quickly about one particular virus in the last few months. But mostly, my lab studies bacterial pathogens. And the one that we study the most is E. coli, which causes urinary tract infection. So to just sort of give these questions some context, and to talk one more slide about virulence mechanisms and what it is that we're really going after. Okay, like what's the level of detail that we really require? What's the kind of understanding that we need in order to understand, develop treatments, and possibly make predictions into the future? Okay, so urinary tract infections, many of you have probably heard of this. I'm sure all of you know someone that has at least had one urinary tract infection. They may not have told you about it, but certainly it's about half of all women at some point in their life have a urinary tract infection. This can be an extremely serious disease. Okay, so here's just a patient. Sometimes a few of us in the field get anonymous. They're not anonymous, but we get emails from patients because they find out that we're researching urinary tract infections, and it's a big problem for them. So here's just a quote from one of these patients. I'm having recurrent urinary tract infections in the bladder for more than 30 years. Okay, and so these patients, they obviously go to the doctor. This patient wasn't just waiting for 30 years suffering. They went to the doctor and they were getting antibiotics. They would get antibiotics every time. And after maybe 3 or 4 or 5 years of this, where they keep going back every few months, it starts to become pretty clear that the antibiotics aren't really helping. And then you fast forward, and if it's going on for 30 years, we really don't understand what's going on here. Okay, so that's the idea. We really don't have a clear or we either don't understand or we don't have the right treatment that's actually treating the key disease. Okay, and so if you just sort of go through this more than 30 years, it causes severe, constant severe bladder pain. It can affect their psychology, right? So it makes me depressed and very pessimistic. It can change their entire lifestyle. So having to live with this debilitating disease for the rest of my life. Okay, so me, my lab, as well as many other labs in the field of urinary tract infection, we've been trying to focus on this problem to try to understand this chronic recurrent infection and try to really provide a new solution for this. And we have made some steps forward, but just to give you a sense of what it is that we're talking about in terms of what do I mean by a virulence mechanism. So here's an iconic image from the field, which this is a scanning, it's a sort of special kind of scanning electron microscope. It's not the standard one. It's a little bit more specialized, so we can see a little bit more detail. What we've got here is up here in the top left corner, hopefully you can see my pointer, up here in the top left corner, this is a corner of an E. coli. Okay, this is a corner of a bacterium, and down here in the bottom right corner, this is an epithelial cell. Okay, and this epithelial cell is inside the bladder. It's the one that's lining the surface of the bladder so that it's protecting the rest of your tissue from the urine. And then you see this thing. You see this thing right here. This sort of long, kind of rod-shaped fiber, and this is a particular protein structure that's made by E. coli. And then this arrow right here points to the key interaction that's happening down here. Okay, and what we've been able to figure out over the past approximately 20-25 years is we know all of the proteins in this fiber. We know how they're regulated inside of E. coli. We know why it is that they're constructed in this way. And the reason is because it's all to put one single protein at the end of that tip. One single protein at the end of that rod. And here's a crystal structure of that protein. And this orange thing right here at the very bottom, this orange thing is a sugar that's on the epithelial cell. And all of these proteins are constructed so that you can bind that E. coli. Okay, you can use these green residues on this one particular protein to bind specifically to this epithelial cell. And that's how the entire infection process starts. One of the reasons that we think this is becoming a recurrent infection is that this particular interaction, this molecular, this sort of atomically specific binding interaction leads to the E. coli going inside of the epithelial cell, and therefore antibiotics cannot reach the E. coli. We can't clear those out. Okay, so this is what I'm talking about, a virulence mechanism. This is the kind of thing that we are looking for, that we're hoping to develop, that we want to understand down to the level of a single gene, down to the level of a single protein, and if possible, down to the level of individual amino acids and their precise atomic interactions with the human host or the human immune system. What's the molecular specificity that allows that bacteria or that virus to cause disease? And once we have that information, once we know what those key interactions are, then of course it becomes very clear. This is the way forward for rational targeting using small molecules to try to either prevent or treat disease. Okay, and so this takes many, many years, of course. And this is sort of what my lab does, my lab does in conjunction with many of the other labs in the UTI field. Okay, so here's just a very brief overview of the kind of activities that are going on in my lab. Of course, as I mentioned, we're studying recurrent urinary tract infections. Larry mentioned that we're also now studying some infectious outbreaks, but the whole goal is to use genomics to try to accelerate this process. Okay, so genomics really has advanced in the last 15, 20 years. And so if we can use genomics, can we accelerate that process to get to those really detailed molecular mechanisms. And not just can we do this for urinary tract infection, but we want to improve the entire process, right? Can we come up with ways using E. coli or using urinary tract infection or using other bacterial systems? Can we come up with ways that then allow us to say, you know, this is a general technique that really would apply to other diseases as well, that we could, you know, accelerate our understanding of other diseases. And of course, that's important because there are some diseases which are difficult to study in the lab, and there's some diseases which are just new. For example, COVID, right? Part of the difficulty that we also have with COVID is we're all building the tools as we go in order to study COVID. What if we had tools that were already applicable from before, right? What if we, you know, had especially computational tools, because all these sequences are already coming in. And so that's one of the huge goals in the lab. One of the major long-term goals is to improve the process of how we actually understand infectious disease and how do we get to in detail particular molecular mechanisms. Okay, so the plan for today, what I want to do is I want to give you an example of how we are trying to accelerate that process. And it all starts again with an individual specific case. And that specific case today that I'm going to talk about is a disease caused by a bacteria called Campylobacter jejuni. It causes abortion in livestock in the US and Europe. And you know, one of the really nice things about this particular infection story is that we were able to get to a single very clear answer. You know, it's sort of like that protein that binds specifically to that epithelial cell. Now we know exactly where we should go look for small molecule inhibitors. Those are the kinds of things that we can turn into an assay and do large-scale screening campaigns. Okay, so it has a single clear answer for Campylobacter jejuni as well, which then gives us a defined path forward for diagnosis and treatment. Okay, but another reason I really like this example of Campylobacter jejuni is that it actually gives us some insights and some hints, some lessons for how we can understand other diseases. And I'll talk about how that led to the development of a potential purely computational method that can actually give us those answers directly. Okay, so let's jump into Campylobacter abortion. So just the postdoc that worked on this in my lab is Bala Periaswamy. We collaborated with Wei Zai, who was a colleague of mine at GIS, and then we also had collaborators at Iowa State University in the US. So a very brief background in Campylobacter jejuni. So I've mentioned a couple of pathogens already. This is another bacteria. The kind of relevant information. So all bacteriologists will always, you know, every time you introduce a bacteria, you have to say it's gram-positive or gram-negative. So Campylobacter is a gram-negative bacterium. This is a scanning EM of this. You may not have heard of Campylobacter, but it's close to Helicobacter, which a lot of people have heard of. You know, in the grand scheme of sort of bacteria, they're somewhat related. And Campylobacter is very commonly known for causing food poisoning. Okay, and here's just some statistics. The food poisoning, though, is because they are frequent colonizers of large livestock and poultry. Okay, and so this is kind of the modern world, right? This is how modern intensive farming works. And I think everybody is very aware of how quickly things can spread in very dense environments like this. This is the whole reason we do social distancing and have to wear masks now for COVID, so that COVID doesn't run through our dense urban centers like this. Okay, but Campylobacter really has a field day when it's coming to these high-intensity farms and these high-intensity modern farms that we have. So Campylobacter normally is an asymptomatic colonizer of sheep, and it can colonize a few different organs within those animals. But then sporadically would cause abortion. And when it causes abortion, so in the pregnant ewes, of course, you would find Campylobacter also in the blood, and you would find it in the placental and fetal unit as well, right? And that's why that infection, that's why it's causing abortion. Okay, so here's just a little bit of epidemiology, and I think again, you know, with COVID, a lot of this stuff, you know, people have sort of seen things like this more frequently now. So Campylobacter, it sporadically causes abortion. Okay, and so if you look at the kind of, these are called sequence types. Essentially, the different colors mean it's a kind of different, say, it's a different strain of Campylobacter, right? It's sort of like how SARS-CoV-2 is different from SARS, and it's different from the bat coronaviruses, it's different from the common cold coronaviruses. It's sort of like delineating these out like this. So these are different. They're all Campylobacters, but they're distinguishably different from each other. And what you see here is it's just a rainbow of colors here, right? The kinds of Campylobacter that cause these abortions in the UK during this time period, it was essentially just any Campylobacter can do this, and this is what you would call kind of a sporadic pattern of causation. If you look at the same time period in the US, this is what this looked like, okay? And you see that this is one color of this thing called ST8, okay? And then here's this rainbow, and this rainbow of all these other sequence types, you know, what it is that this small piece of the pie is actually as big as this piece of the pie here in the UK. It's just that ST8 caused an excess of abortion in the US, and it was all caused by this single strain. And of course, we can see that on the genomic level. It's all these red ones. So again, I'll go over these phylogenetic trees in a little bit more detail later, but suffice to say, at this point, I think, you know, again, COVID has sort of done a lot of the pre-education on this in terms of what these sort of family trees of viruses versus bacteria, what they look like. And essentially, these guys are all very, very, very closely related. So this is a clonal outbreak, a clonal epidemic of abortion that's happening with Campylobacter. Okay, so genomics has been around for a long time, and you know, it's made a lot of advances over the past 15, 20 years, as I mentioned. And so genomics has been something that we've always wanted to apply. We've always wanted to apply to pathogenesis or to infection. And so one of the things that was done, this was about 7 to 10 years ago, is if you look at this family tree of Campylobacter, okay, so this IA3902 down here at the bottom right, IA3902, this is one that causes abortion. And they actually happen to be a closely related Campylobacter, this NCTC11168. This Campylobacter, while it was closely related to this IA3902, it did not cause abortion. Okay, and then that actually was backed up by animal experimental data, where you could take these strains into the lab, you could infect pregnant guinea pigs, and IA3902 would cause abortion in those pregnant guinea pigs, whereas NCTC11168 would not. Okay, and so this made sense. So now we can take these two and try to compare. And we say, what are the sequence differences? What are the differences in the genetic material that might then tell us why IA3902 is more virulent, right? Why does it cause abortion? And if you look, if you sort of sequence these genomes, and you've got 72 genes, there's 8,000 SNPs. So SNPs are single nucleotide polymorphisms. These are, you know, these single nucleotide polymorphisms, those are the kind of thing that was in that news article or that preprint about the more virulent form of coronavirus. They're talking about one single nucleotide difference, okay? And now just between these two, we've got 8,000 of them. And all of them potentially could cause a difference in disease. There's also a plasmid and then a difference in antibiotic resistance. Okay, and then as you look at these, as you sort of go through these 72 genes and you look at these 8,000 single nucleotide polymorphisms, you can try to say, okay, well, what are the functions of these genes? Right? Does that tell me something? Because just having 8,000 differences, okay, I don't really know what that means. But maybe if I find differences in a gene that I know is important, right? If I find a difference in antibiotic resistance, or I find a difference in, you know, I don't know, motility or something, then maybe I can use that to figure it out. And so if you go through that process and try to look at the functions, you come up with a bunch of these hypotheses, right? So motility. So bacteria that can swim a little bit faster, that seems to be important for them to either get through mucus to get to the epithelial cells and cause disease. Capsule is another one. Capsule is something it's this sort of sugar coating on the outside of bacteria that some bacteria have, and it can actually block the immune system. It can prevent immune cells from killing off the bacteria. So that's important for disease as well. Iron acquisition, that's another system that, you know, basically mammalian hosts will limit iron. This is called nutritional immunity. And so bacteria that bacteria will starve for iron. So the ones that can acquire iron better, they have specific genetic systems that allow them to sop up iron. Those guys do better, et cetera, et cetera, et cetera. So all these things, right? These are all reasonable hypotheses. These are all things that we know can be important for bacteria to cause disease. Okay, and the problem is all these are hypotheses. There's lots of question marks here. You notice that every time I'm going through one of these, I say, this is something that is possible for bacteria. It's possible to help a bacteria become infectious. Okay, and really what that is just like this. All of this, these hypotheses up here were no different from what happened with this article here, which is that you say, I see a sequence difference. I'm just going to, it's certainly a possibility that that sequence difference is important. Okay, but as you know, Ed Young sort of noted in his article, whether or not that's true, we just don't know, and it's very difficult for us to act on that data because it could be a false positive result as well. Okay, so this is the fundamental conflict or the fundamental problem we have. And so the whole goal, of course, is let's do better, right? So we've had many years of genomics, we've had many years of infections, we've had many years of experience. Can we actually do something better than this? And you know, to try to figure out a way to move forward, one of the first things we did was my collaborators in Iowa State and I, we sort of sat down and sort of tried to think this problem through. What are the advantages that we have? What are the assets that we can use? And I mentioned to you already that, you know, one of the things that we don't have for the SARS-CoV-2 yet, right? One of the things that we haven't been able to really, not to the level where we can differentiate virulence, right? We don't have an animal model for that infection, but we did for Campylobacter. We could actually study that in the lab. Okay, so we have two strains, one causes abortion, one doesn't cause abortion. Now we can use those to try to dissect what the relevant sequence differences are. So we have a good guinea pig model. We have an animal model we can use in the lab. We have a closely related non-pathogenic strain that we can sort of do the comparisons with. And then there's a third thing, which is kind of specific to Campylobacter. It's something that's shared by only a few different bacteria, and that's something called natural competence. So natural competence, you may remember from maybe university biology that there's, you know, bacteria can acquire extracellular DNA. They can acquire foreign genetic material, and there's a few different ways that they can pick up DNA from the outside, and competence is one of the ways. Competence is, you know, you might have heard of transformation, transduction, and so competence is one way that they can do transformation. And what it is you have a bacteria, and it's color coded here. So the original chromosome is red here, or sort of this sort of brownish red maroon. And then when there's DNA on the outside, and they just color code this blue so that they color code this blue so you can keep track of it. This bacteria, Campylobacter, can just directly take up that DNA and then incorporate it into its chromosome. And this incorporation process is exactly like how we do sexual genetics in humans or plants or mice. It's a homologous crossover. Okay, so it's like exchanging alleles. It's like having two parents, and so this is why sometimes this process is called bacterial sex. Okay, so we realized that with these advantages, we could come up with a kind of, you know, at the time was a very sort of crazy idea, which was what we're going to do is we're going to take this non-abortion strain, and I've color-coded the chromosomes here again. So this represents one bacteria, and the black squiggly line inside represents the genome. So this non-abortion causing bacteria, we're going to just inject free DNA. So we're going to purify DNA from the abortion causing bacteria, and that's color-coded in red. Okay, and then we're going to rely on this natural competence process here, where now this extracellular DNA will just get randomly incorporated through homologous recombination. It will get randomly incorporated into the non-abortion strain. And so different pieces of the chromosome will get incorporated into different clones of this non-abortion strain. And if it just so happens that one of these pieces of DNA that gets incorporated, if one of these pieces carries the key genetic change that's required to cause disease, then this bacteria, whatever bacteria that is, should now be able to cause abortion. And we can test for that using our guinea pig model. And then, of course, now we can just sort of do this all in one experiment and sort out all the details by sequencing. Okay, and essentially this happened very quickly once this idea finally got going, and we sort of worked out some of the details as to how to do this hybridization in vitro. In the very first animal experiment, okay, this is what we saw. And so you've got your positive and negative controls here. So this is this line up here. This is a standard survival curve, but the survival curve at this point it's a survival rate for the fetuses. Okay, so we're talking about the abortion phenotype here. So this is the non-abortion strain, and there's no abortion. When you put in IA3902, so that's the circles, you see this abortion occur very fast. And when you infect those guinea pigs with this pool of hybrids, right? So you're starting with this one that doesn't cause abortion up here, and then all of a sudden you get some abortion. And you can maybe see that a little bit more clearly here if you look at the bacterial levels. So the y-axis here is higher bars or more bacteria, right? And so we're looking in both the uterus and the placenta. Of course, this is where, you know, the infection is happening, and that's really responsible for that abortion. Again, NCTC, this is the one that does not cause abortion. If we just sort of focus on the left one here, so NCTC does not cause abortion, and there's no bacteria in the uterus. IA3902 does cause abortion. You see these very high levels of bacteria, and then those transformants also can get into the uterus and the placenta and cause an infection there. Okay, so amazingly, this experiment worked. We've been able to transfer a piece, or we think we've been able to transfer a piece of DNA, right? We think we've been able to transfer a red piece of DNA into this non-abortion strain that now it can cause abortion. So what we're going to do is we're just going to take these bacteria that were in the uterus and placenta, and we're going to sequence them. Okay, and then just see what DNA got transferred, because we have the genome sequences of both the parental strains of both IA3902 as well as NCTC 1168. We can see which piece of DNA came from which parent. And here's a graph where the columns, each column is a different strain that we isolated from the uterus or placenta, okay? So we have 12 different strains here in the columns, and each row here is a different gene that may or may not have been transferred. If it's colored red, that means it came from the abortion strain. If it's colored blue, that means it's the original NCTC, the original sort of non-abortion version of it. And what we're looking for, of course, is red all the way across, because that means that that piece of DNA was commonly transferred in all the abortion-causing strains. And so if you look, it's pretty quick. You can just see this red block up here. They've sort of all been ordered at the top. It turns out that ends up being 7 genes. Okay, and remember, we started with 565 genes that we were looking at, but now we're down to 7 genes. And we looked at that sequence very closely. 5 of those genes had errors in the published non-abortion sequence, so actually we could eliminate them. They actually weren't transferred. There's no difference. And at the end of the day, of the 2 remaining genes, only one of them had any amino acid changes. So one had some sequence differences, but it didn't change the amino acids. So it ended up being that we had 1 single gene. Okay, after a single experiment in guinea pigs, we had a single gene after sequencing. And that gene is called porA, and then we went back and did the genetics. So what we did was we took IA3902 and we took porA from NCTC 1168 and put it into IA3902. Okay, so just be clear, porA is present in both IA3902 and it's present in NCTC 1168, but the sequence is different. Okay, so we can say there's an IA3902 version and a NCTC 168 version. And if we swap the versions between these 2 bacteria, that's what we're doing here in this experiment. Okay, and then actually maybe it's a little bit easier to look over here, that as long as the porA is NCTC, you have no bacteria in the uterus and there's no abortion. It doesn't matter if the rest of the chromosome is NCTC 1168 or IA3902, so long as this one gene, porA, has an N here for NCTC 1168, it does not cause abortion, and vice versa. If just that one porA gene is the IA3902, the whole rest of the chromosome can be NCTC 1168, and it causes abortion just as well as the original IA3902 strain. Okay, and so for the kind of, you know, the sort of geneticists out there, is essentially the necessary and sufficient experiment. This is really the one thing that is truly important for causing this abortion phenotype. Okay, and so of course now this gives us some tools that we can use for diagnosis, for screening. This gives us tools that we can use to follow up and say, what are the assays that we need to do to develop small molecule inhibitors of whatever it is porA is doing? Or we can focus on porA and say, is this contributing to invasion, or is it contributing to immune evasion? Okay, so here's the opportunity, right? On the left-hand side, this is what we were able to do before we did this, hybridization plus sequencing strategy. And then on the right-hand side, when we did that hybridization strategy, which we call sexual genetics and bacteria, we were able to get one clear gene, okay? And this is essentially this difference, right, between speculation on this might be possible because, we see this DNA sequence difference that might be related, and so, this could explain what's happening, versus, what? We've got the gene, we've got the structure, we've got the interaction, we know what test to run, and we know how to block it. Okay, and this is what we're looking for definitive mechanisms of virulence. Okay, so this is the opportunity. This is something we're able to do in Campylobacter. But how can we sort of extend that to be more usable outside of Campylobacter? How can we extend that to be useful for other diseases? And so one of the next questions we asked was, in Campylobacter, we've got a bunch of DNA sequences, and the DNA sequences before, since we didn't really know how to analyze the sequences, we were coming up with these kinds of answers. We'd like to be able to say, can we take those DNA sequences and come up with this answer here on the right-hand side? Okay, and this is what I call reverse genomics. And I hope you can see that if we can come up with a computational method to just analyze sequence that takes us from genome sequence down to porA in a single step, that would qualitatively, that would be a quantum jump. It will be a quantum improvement in our ability to get and accelerate genomics answers for infection. Okay, so how do we go about doing that? Well, we had to develop a model in terms of what's really happening with Campylobacter. How is it that abortion strain evolved? And the thinking was that it acquired some mutation that, it essentially acquired that porA mutation that enabled it to cause the abortion phenotype. And then now it had that abortion thing, it could become more successful, and it started to rapidly expand. It was spreading through those entire flocks of sheep and causing, it was really a bad problem economically. Like as soon as one ewe would have abortion, the farmers just knew that every other ewe for that entire season, every other one would be aborting their fetuses. It was really bad. Okay, and so this is this pattern of like you have one mutation, or you have the key mutation that we really are interested in, and then you have this big expansion. And so it turns out that there's been a lot of theory developed. There's a lot of techniques that might be usable to try to understand when there's a population expansion, or when you have a selective sweep, or, is there something to do with selection? Okay, and so we tried to make as much as we could, we did a complete list of all these things, and just essentially said which of these can detect porA out of these Campylobacter genomes. And it turns out only one of them detects porA, and it works really well. And so this analysis is called FUSFS. It doesn't really matter what the name of this is. It's just some statistic that we can calculate based on DNA sequence. And so what I'm showing you here is genome coordinate. Campylobacter has a 1.6 million base pair genome. So that's the entire genome. Each of these dots essentially is like one gene. And what we're doing is we're calculating FUSFS for a single gene, and there's about 1,000-something genes here that we're calculating. And the two red points, these are the ones that are highlighted here. Okay, so these are windows that are overlapping porA, and you can see that these break the distribution on the bottom side, which is what you would predict if they were responsible for this population expansion. Okay, so FUSFS, like the mutation that occurs here, the theory is that FUSFS should be negative if you look at that gene. Okay, and that's exactly what we see, and it breaks the curve on the bottom. Okay, so this is where we were with Campylobacter. We were able to combine biology and genomics, sort of as Larry said. This is, I think, where a lot of power comes from. That if you sort of deeply understand the biology, as well as be able to sort of apply some new genomics techniques, those really can build and leverage off each other. And so we did that for Campylobacter. It's a technique that we call sexual genetics. And then that enabled us in essentially a single experiment to identify a single gene that's responsible for sheep abortion, something that had eluded studies for the previous, say, 10 plus years using other kinds of genomic techniques. The second lesson is after we know that porA was the important gene for Campylobacter, we could actually use that to try to discover a new computational technique, or to discover, really to learn how to do the computational analysis better for the next time. And what we learned is that an infection outbreak is a very distinct event. An infection outbreak leads to a rapid expansion of whatever's causing that disease, whatever's causing that outbreak. And so if we can find techniques, find analyses that can detect population expansion, this may be a general way that we can accelerate our understanding of disease, even if we don't have an animal model to work with in the lab. Okay? And so I'm going to switch to another bacteria that, is something that Larry alluded to as well, and represents that kind of thing where we don't have an animal model. This is a totally new disease. And can we try to apply some of these technologies, some of these techniques, or at least some of this thought process to this? Okay, so this next section is about this outbreak of foodborne group B Streptococcus. And you're going to go back to kind of popular media here to really, drive home this point that many things are happening. There's always new infections coming up. COVID-19 is a totally new infection, but it's not the only one. These things are happening all the time. So this is a WhatsApp message, and this is on 12 July in 2015. Okay, and so this might be a little bit small, but I'll just sort of summarize some of the key points. It's a WhatsApp message that just got shared all around. So in case you don't know, I don't know if you guys use WhatsApp so much in Qatar, but WhatsApp is essentially, it's kind of like one of these text messaging systems, right? It's a little bit like WeChat. It's functionally the same as WeChat or Facebook Messenger or something like this, or like an evolution of SMS. And so this got forwarded to many people in Singapore. And it says, I'm sharing this because there's somebody, NUH is a hospital, so somebody in the hospital because of a painful right arm and ate raw fish at one of these. So this is just it was just a particular location here in Singapore that has these food stalls. So ate raw fish and then gets into this. There's a surge of young and old men who've been coming in sick, and it's a particular strain of bacteria, group B Streptococcus, and it's associated with this raw fish. Okay, so this was 5 years ago, right? But if you kind of rewind and go back to what did we know in 2015 about this bacteria, okay? This WhatsApp message was going around so fast, okay? And here's the big problem. It's new. What do you do? And the Ministry of Health had to make a response. So you can sort of see in this news article, they're referencing this WhatsApp message, and it's coming a day later because it's just going through the entire population. And then the statement was no link between eating raw fish and GBS disease. Okay, and actually that's a true statement. At that time, there was no evidence, absolutely none, that you could get GBS disease by eating raw fish. Okay, that's just true. There's no evidence. Doesn't mean it can't happen, right? So, and you look at the dates on these. So the next day later, right, it became very clear that there's a very strong association. Now we have a little bit of data, and so then they I have to say it's a very difficult job. I'm not trying to sort of cast any criticism here. It's a very, very difficult job when you have a new disease that's coming out. We saw this with COVID-19 as well. Okay, the Singapore MOH turned around very quickly as soon as they saw this data. Okay, and then they got right on it, and I think they did an admirable job, because the next article is that they clamped down on the raw fish dishes and the outbreak went down. Okay, so this is the environment we're in. Things happen very fast. News can spread very fast, even if we don't know what's really true. Okay, and actually maybe news spreads faster if you don't know what's really true. Okay, and that puts us in a very difficult situation. And this is one of the main reasons why we want to accelerate the ability to find these answers. So we can help MOH, we can help doctors, we can help the general public to have better information faster that you can rely on. Okay, so just to summarize this part here, group B Streptococcus, it's a gram-positive bacteria. I told you, as a bacteriologist, I'm always going to tell you gram-positive or gram-negative first. Okay, so it's a gram-positive bacteria. Here's a scanning EM that's been artistically colored in. It's a very common bacteria. It's present in 30% of healthy adults. It does cause a lot of disease in very young children, so neonates, right? Directly after birth, within the first seven days of life, it can cause meningitis. GBS had never been associated with food. Okay, so this is a bit strange, and that's, of course, this is why you're seeing those articles saying there's no link with eating raw fish, right? Because GBS had never been associated with food, no one ever looks for GBS in food, so it just seemed like a very strange thing. And people didn't really believe us, or people didn't really believe Singapore actually at the beginning. Okay, but so here's an epidemic curve. I'm just going to skip this. I mean, it basically just sort of says that as soon as they figured it out, the outbreak went down. Okay, and in terms of this idea that it's a new infection and there's just a lot of skepticism. So the particular bacteria, again, I mentioned these sequence types of 4 for Campylobacter, or, it's sort of like coronaviruses. You can say coronavirus, but they're different kinds of coronavirus. So for GBS, there's also different kinds of GBS. And this particular GBS that caused the outbreak in Singapore, it's called ST283. Okay, so it's just a particular kind of GBS. And it had been reported in the literature before. There was an invasive infection as far back as 1993. There were some other strains that were very closely related in Singapore in 98. Okay, but then none of these studies had ever really understood what the disease was or how it was transmitted. It was very rare reports of ST283. Okay, so this was the question that we got, especially for some of the first papers. Is the outbreak, really, is this? We know group B strep, it causes these disease in babies. It doesn't cause disease from eating raw fish. Okay, and so I'm going to skip over some of the other pieces of data that went into this, because the part that we did was to help with the genomics. Okay, but just to be very clear, the genomics was only one piece of the entire data package that went into arguing that really, truly, this is a foodborne illness. This really was associated with the raw fish. So this is like a family tree of group B Streptococcus. Okay, there's about 500 strains in here, and you read this just like a family tree, except it's sort of turned on its side. So if you were to look at these sort of branches here, like or if you look at these red stars here, these are like twins or siblings or something. Whereas then if you look at these guys, it's like these guys are totally unrelated. Maybe they're like related 12 generations back, right? You have to go back to the parents and grandparents and great-grandparents, and that's how they're related, that finally you get down to these descendants, and they're just further apart on the family tree. Okay, so the kind of closer that they are, on these branches, the more closely related they are. And you can really think of it as kind of like a family tree. And so these outbreak isolates, right, we saw that they were all clustered here and they look very, very close. And if we zoom in, then I zoomed in about 1,000 times here, and you can see that this line is still vertical. The little branch links here are so short you still can't see them here. So I'm going to zoom in another 1,000 times, and now you can finally start to see some of this differentiation. And again, I think, hopefully you've seen some of these and know how to interpret these based, like you'll see these in the newspaper articles these days with these phylogenetic trees. Again, what we're looking at is these very short branches. This is one nucleotide difference, one SNP difference out of 2 million. Okay, and you can sort of see that the outbreak that we had was this pink section here in 2015. They're all from Singapore, but then we also saw that this strain was around in previous years in Singapore as well. But the key piece of information right here is where did we get these isolates from? And you see this one. This is all blue here. Those are human cases that were sick. And if you focus on 2015, okay, so here are all these guys, and there's one of these that's yellow. Okay, and it's this particular one because this particular SGM1, this particular isolate of group B Streptococcus, the same DNA sequence was isolated from patients as well as fish samples at the same time. We could not tell them apart. It's like having the exact same fingerprint at the scene of the crime, right? It was exactly the same. And these numbers in parentheses here is 72. So in fact, what I've done here is I've collapsed all of the identical sequences. So this actually represents 62 humans and 10 fish samples all had the exact same strain of group B Streptococcus. Okay, and again, there are other pieces of data on the clinical side and on the epidemiology side that fit in with this genomics data to make the complete picture that it is causing this outbreak. Okay, but this is just the one piece of genomic data that was very, very clear that these were clearly coming from the fish and going into the humans. Okay, and then we could sort of do some additional analysis. This is some of the stuff that my lab does where we can try to infer when was this happening. And ST283, the one that caused this outbreak, seemed to emerge in 1985. Okay, that's very, very recent. Okay, so, if you think of, okay, it's not recent like COVID, which is, 2019, okay? But many of the diseases we have, many of these bacteria, we know them if we do the same kind of analysis, they've been with humanity for tens of thousands of years. Okay, so this is a newly evolved clone of group B Streptococcus just in the last 30, 35 years, okay, that's now causing this new disease. Okay, so in summary, GBS causes foodborne infection is totally new and we aren't sure why. Okay, so now I'm going to tie this back to how do we actually figure this out? So we know that it's associated with fish, but that still doesn't give us a great way to go after therapeutics. And how do we apply some of the tools? How do we apply some of the tools that we learned from Campylobacter to try to drive towards a deeper understanding of what happened in and about 1985 that led to the emergence of this disease? Okay, and I have to say, that's the goal, and we don't have an answer yet, but I have some preliminary results that I'm going to show you here that I think show some promise. Okay, so we don't actually, we don't actually have the answer to that yet, but remember I had this model where when you have an infection, it's a rapid expansion, okay, of a single clone, and we're looking for maybe a mutation that's happening here. Well, group B Streptococcus, it has a lot of rapidly expanding clones. And the ST283, it turns out we don't have enough data for that yet. Okay, we've only had like one outbreak, and it was sort of taken care of relatively quickly. Okay, but there are other clones of group B Streptococcus that are causing disease, and we have lots of isolates of these. So we took a look at SS1, this one up here. Okay, and if we look and we use that same FOOS-FS technique, okay, so again, this is the same thing. Group B Streptococcus has a genome that's a little bit longer. It's about 2.1, 2.2 megabases. Again, each dot represents like the FOOS-FS score for one gene. And what you're looking for is FOOS-FS scores that, come down a little bit lower. And what we see is there's one screamingly hot screaming signal that's down here for one gene that's a fibronectin binding protein. Okay, and this is, again, it's one of these things that we have always thought that fibronectin binding proteins might be involved in pathogenesis, but we don't have any data for that yet. Okay, and again, this is still at this point just a prediction, but this is something we're trying to follow up, again, using experimental validation in the lab to really figure out, does FOOS-FS work for these other bacteria as well, not just for Campylobacter? But in the meantime, we can still run that computational technique on everything. We find different answers for different clones, which is exactly what we would expect. And I should say that this looks like it's the same answer, fibronectin binding, but they're two different genes that we got the signal for SS1 versus COH1. Okay, so then just to sum up, right, so I sort of covered a little bit of this before, that the sexual genetics, the combination of biology and genomics is extremely powerful. But then, using this idea that infection outbreaks are a distinct event that lead to a population expansion, this may actually work in multiple bacteria. And there's no reason that it should only work in bacteria. It should also work for any other organism that's causing a disease and rapidly expanding, such as, viruses or viruses like COVID. Okay, so virulence mechanisms are still hard. This is the last slide I want to sort of sum up with. But these new methods are showing some promise. So what I didn't talk about today was some of our work trying to use sexual genetics in other bacteria, not just in Campylobacter. But that's an experimental thing. And I think one of the things that's really has the potential to increase our ability to accelerate our ability to understand these virulence mechanisms are these computational tools that work only requiring genomic sequences, because we're just getting so many of these now, and we just don't have good ways to analyze these sequences. Okay, and so I'd like to acknowledge the people in my lab who really do most of the work, and or sort of the greatest joy that I have in running a lab is really seeing them grow. And so I've bolded the people who were involved in the projects here. Kurosh was involved in the Group B Strep project, and Bala I mentioned for the Campylobacter project. And then collaborators, funding agencies, and support. And we're online. I can't travel to Qatar right now. You guys can't come to Singapore, but we can share some pictures. And this is actually, Singapore doesn't look like this right now because there's too many people here. But Singapore is really a great place, and when travel restrictions lift, hopefully you guys can come out here and visit at some point as well. Please look me up. Thank you very much.
Swain, thanks a lot. I'm sure we have lots of applause. Fantastic talk. And I do have some questions that have come in, and I just maybe remind the audience that if you want to post some questions, I'll try and consolidate those and take advantage of some time that we have to pose a few questions to our speaker. So again, thanks, Swain. It is very successful. Just so you know that we have about 50 attendees live, so that's really fantastic. And really excited about that. So maybe I'll kick off with a question that I have from sort of the first part of your talk, where you did this nice study and came up with porA as a necessary and sufficient gene. And I think a lot of us do these sorts of what's called functional genomics type projects. We want to find the gene, we want to publish what the gene causes the disease, and I'm not sure I'm disagreeing with you, but I'll just quibble on the sufficiency issue, because obviously, you're putting this genetic transfer into a background that maybe is already primed to do the infection. So there's other gene-gene interactions that are going on. Certainly, porA is critical. And I know you think about this, and all of us who think of complex human diseases, they're multigenic. Rather, what is your thinking on how you can broaden this, not to only look at single genes and maybe even solve sort of secondary events in your FS analysis or something? Can you start looking at multigenic, more complicated scenarios? Obviously, it's somewhat dependent on both the genomic and computational tools, as well as the biological system you have to interrogate your hypotheses.
Yeah. Okay. So that's a great question, and for sure the claim of sufficiency is that is context dependent, but so to sort of focus on that first, like we did actually just move that gene, and that's the only gene we moved over, and it's between those two strains. Okay, and those two strains, admittedly, are very, very, very closely related to begin with. Like as far as Campylobacters go, they're pretty close already. So yes, I'm sure they share, there's all sorts of stuff that these bacteria do already. They can evade the immune system. They can survive neutrophils. They can, they can actually acquire iron, stuff like this. Okay, so, but so you could say that the exact experiment that I showed and that we did is kind of a limited context, but in that sense, okay, like it's like doing an exogenous sort of allele swap. But there was a natural experiment done. So this is other data that as we were looking at genome sequences, and now that we knew what that allele was, so Campylobacter actually does a lot of recombination in terms of it exchanges DNA between different strains a lot, and that's related to the fact that it's naturally competent. And it turns out that it just so happened that we caught two strains, okay? And one of the strains was genomically, if you draw a whole genome tree, it's right in the middle of the Campylobacter of the abortion strains. But it clearly has a recombination. It clearly has a different allele of porA that's been recombined in. And it's very different, right? So all the ones that cause abortion, they might have 1 or 2 mutations away from each other. This one's got 60 mutations away. And that one was the only one that was not isolated from an abortion case. Okay, so, we still didn't do the experiment to sort of prove that that didn't cause abortion. Okay, but that's the first one that backs up the kind of necessary thing. And then the second, just totally serendipitous observation was they caught another strain that was, because they're always screening for abortion isolates, they caught another strain that was completely different genomically completely different genomically from that epidemic clone, but had clearly recombined the exact porA from that epidemic clone, and that was isolated from a an abortion case. So there's a natural version of this that then went even further away genetically than we did. So I think, I agree with you that like there still has to be some couching of, what we really mean by sufficiency. But it's, like we can't sort of put that in E. coli and have it cause abortion, for example, right? It's not gonna work like that. But I think within Campylobacter, the data is looking pretty good that those are the mutations that are actually required. And then, sort of somewhat quickly on your second point about are there multigenic, are there sort of multiple things that are important for virulence? And this clearly, we think that there must be, right? You must, as I mentioned, you must have to already be able to grow. You have to be able to avoid the immune system. You have to be able to, like actually outcompete other Campylobacters or other bacteria wherever you're going. And so that's true, but I think one of the things that we're seeing, and this gets to the sort of predictive power of understanding how diseases work. The thing that we're seeing with FUS-FS is that we're catching perhaps the last mutation that was required to get to the point where you can cause disease, right? So sure, there's all these other things that this bacteria's got to be able to do, right? But it ends up being, for Campylobacter, the data seems pretty clear that it was only one last step. And we actually think that that's generally the case for many of these bacteria. That's one last step that they need, just that one last thing, and then they can cross that epithelial barrier. And that's what we're really looking for, So I think it probably in more cases than we expect for bacteria, at least, it's actually just going to be one gene. Yeah. Not all the time, but for more cases, yeah.
That's a fair answer. There's some other nice questions that have come in that, one is, I mean, you sort of in the lab, created this, the sexual exchange of nucleic acid into the strain, to discover, but, in the natural world, and you kind of touched on this a little bit, there's obviously mechanisms for which nucleic acid gets exchanged between substrains and colonies. The one question wanted to know, what kind of evidence there are, are there bacteriophages that are moving nucleic acid between, let's say virulent, making them virulent? Maybe you just touch a little bit on bacteriophages.
Yeah, for sure.
Other mechanisms of nucleic acid exchange.
Yeah, that's definitively happening, and there's actually, the more we look, the more mechanisms they are. So, I guess like the classic way that bacteriophage would exchange DNA would be, okay, you just have a phage, and it carries not just the phage genes, but it can carry accessory genes that may be responsible for antibiotic resistance or some virulence functions, and so those just get packaged along with the phage and can move from genome to genome. We also know about things like specialized or generalized transduction. So these are sort of classical mechanisms. And so that can transfer any part of the genome. But one of the really interesting things, so it's a colleague here at NUS, so he's a young assistant professor that just joined about 3 or 4 years ago. So they had a recent paper where they're showing yet another mechanism whereby phage in Staphylococcus aureus, phage are transferring huge sections of the genome at very high frequencies, like a million times higher than we ever expected in the past, right? And it actually ends up being one of the most efficient ways for Staph aureus to exchange DNA is these it's not actually phage DNA that's getting transferred. It's regular genomic DNA, but the way that it works is it's some site similarity between the packaging signals for the phage. And so there certainly are many ways that DNA can get transferred, and we keep discovering them. Bacteriophage, probably, like one, we already know about mechanisms they do, and there are probably other mechanisms that they can mediate that we also don't know about as well. But for sure, they can do this, for sure.
Great. Let me relay a question. Maybe you'll have to interpret it as well, because I'm not quite sure what. It's actually from somebody in Nigeria. I hope he's tuning in and watching live. So he's asking, in the case of abortion, what are likely infection diseases rise to? Here in Nigeria, there's a high rate of abortion, but I've yet haven't seen any research proof any infection possibility. I don't know if he's talking livestock or if in people,
Yeah. Okay, I mean, okay, then in that case, maybe we'll just limit it to livestock if it's not specified. I think in livestock, because that stuff is, it's a little bit easier to kind of track livestock. And so like, I'm not a veterinary or agricultural person, really. We're sort of helping out with some of the genomics, but I think, It's okay. Maybe I should be very careful here. I don't actually know what the proportion of infectious ones are, but I do know that all the infectious ones they capture, or at least like the systems I'm familiar with in the U.S., because those are economically important. They're looking out for these kinds of epidemic transmissible forms of abortion. So they have national labs that are collecting all these isolates. But I'm afraid, like, I'm not also 100% sure what that question, like, if it is supposed to be about humans as well. So maybe it shouldn't
Yeah.
spend, it shouldn't answer the wrong question here.
So let me ask, this is sort of maybe a broad question in the sense of, your work on the GSB was really, fantastic. And, one thing that I've always been amazed at is that, even though we're starting to marry, the clinical work, the epidemiology work with genomics, I just feel like it hasn't caught on fast enough. I mean, what is it going to take, that we can start surveying, these potential pathogenic, bacteria and viruses, and be ready for when they emerge. Do you maybe this is sort of thinking WHO level? I mean, are we
Hmm.
beginning to see some buy-in to the ability to use these tools and not react after COVID type things happen,
Yeah.
but to be ready to say,
No, that's
we should
a
be ready for
yeah.
coronaviruses, and a coronavirus is going to jump, or we should be ready for a jejuni that is going to jump.
Hmm.
So I know this is a pretty broad question, but maybe you can just comment based on what
No,
you're seeing.
I think that's a critical question because and this as well, right? We've been beating the genomics drum for years. And genomics has made a huge amount of progress, right? So there's these graphs that say genomics is progressing, the cost is coming down faster than Moore's law, right? Moore's law was the one, this exponential rate of progress that took computers from warehouses into your pockets, or now on your wrist, right? And we're going faster than that in genomics. Like, why are we still reacting to this, right? And I would say that that rate of acceleration, that rate of progress is totally true. We're going faster. And so the thing is, we're going to get there. But I think at this point, it's still expensive. I think really it's a cost thing. There's also an analytics problem. But I think, like, once the data is there, then the analytics will come, right? There's no sense in making the analytics if you don't have the data set that's sort of coming in live. But I would say that it's more like, if you go back and rewind, say, like 20, 30 years, again, looking at computers, you would say, oh my gosh, the computer technology is so amazing. Right. And you can actually go back and take a look at some of the early days of the internet where they could sort of see the potential, right? But it still took another 20 years of progress, 20 years of improvements, 20 years of cost coming down before we really got to this point where we can really carry these devices and have internet. Like, there are always the early adopters that wanted to have internet, like, in the carried around with them in the 1990s or the 2000s or something, but it just wasn't there yet. And I think that we're in a similar kind of adolescent phase in genomics, that we can see the potential. We've seen so much progress already, but really there's going to be another 3 orders, 4 orders of magnitude of progress, decrease in cost, and that's really going to break open these kind of preemptive kind of surveillance strategies, because right now it's still a little bit too expensive.
Yeah. So your buddy PK had a question, as you might expect. He wants to
Mhm.
know specific residues and structural changes in the porA. Do you know the specific residue? And if you have
Yeah, I can't quote the specific residues, but so porA is an outer membrane protein. Okay, and the uh, based on sequence homology, it appears to be, so a lot of the gram-negative bacteria, they have these outer membrane proteins. So this appears to be like the major outer membrane porin in Campylobacter. And so that would be most closely related to OMP-C and OMP-F. And, based on sequence, you can sort of thread that structure. And so OMP-C and OMP-F, those are these beta barrel, I think they're beta barrel proteins with the beta strands traversing the membrane, and you've got these extracellular loops. And those extracellular loops are really important for kind of modulating function and pore size. Those are also subjected to immune selection. Okay, so they're the ones that are on the outside, and they're antigenic. And so it turns out that all the extracellular loops on porA as you might expect, or seem to be highly variable because they're under immune selection. But then it ends up only being one loop, and I mean, I can just sort of say it's loop 4. It doesn't really matter, but it's between these two beta strands. There's one loop where there's about 5 or 6 amino acids that have been changed. And actually, our collaborators did a couple more detailed experiments where they transferred individual loops between NCTC 1168 and IA3902 and narrowed it down to just loop 4. Just those mutations on that one extracellular loop are able to confer the abortion phenotype on NCTC 1168. So so that narrows it down to a space of about 10 amino acids or something like that. And then it's extracellular, so it's a reasonable hypothesis that it's some kind of interaction with some host protein. I mean, that's the thought at this point, but they're still working on that actively.
Right. So I want to ask another question from my colleague, Dr. Shariar at QBRI, and interested in your use of the FUSFS in your talk. But how about applying that to cancer genomes and maybe other disease-associated genes? I mean, is that something that, or maybe some of those other related computational tools that you were talking about?
Yeah, so we've thought about this, and I think. Okay, so the quick answer is we could apply it to anything, right? It's just we just need DNA sequence and we could just do it. So I'll tell you that we already did this for this ST283 GBS, and that's why I can tell you that we just don't have enough data. So I think again, this was in my view, from a bacterial genomics point of view, this is the first time we've had a technique like this that could really just go straight from genomes down to a single gene and actually mean something. Okay, so I certainly hope that that's not going to be the only technique we ever have, right? That this is just the beginning. And, why would we need other techniques? And I think it's going to be, just like we can't apply FUSFS to ST283 because it's quote unquote too young. Like it's too early. We don't have enough mutations that have accumulated to sort of see the variation patterns. Conversely, you might have things that are quote unquote too old, right? That, maybe tuberculosis has been around for 20,000 years and a lot of these signatures have gotten erased. Campylobacter seems to be kind of in an intermediate stage. And so maybe there's a sweet spot for FUSFS, and so hopefully we'll have other techniques. Okay, but so we've applied.
And somebody else commented that as well. I mean, do you have enough sequences? Enough.
That's exactly right. And that's exactly right. And I think for cancer, so it's a sort of specifically address cancer. One of the issues is if you go inside a tumor, I think you're not going to have enough data because the rate of mutation that we need to sort of really narrow down genomic windows, you kind of need a lot of data, right? And now if you try to take samples across different people across different tumors, now there's potentially a lot of heterogeneity in terms of mechanisms, right? And so, and then also you have the difference between individuals, the genetic difference. And so that's something, again, that we don't have that issue with bacteria because these are generally clonal expansions that we're working on at this point. Okay, so at this point, it's not a great fit. I could see how in the future, though, again, maybe some other different techniques will be applicable to cancer, though. And that's a big hope, of course. I do think there are going to be more.
And just kind of continue on that, there's several questions from multiple people, kind of we all have COVID on the brain, and so we're all thinking about it. So, when you start presenting, this sort of, genetic information, and you kind of touched on that in your talk, you saw all these SNPs, across the, the SARS genomes, and it's, SARS-1, SARS-2, there's other coronaviruses out there, people want you to figure this out, Swain.
Well, so SARS is a very COVID is a very similar situation to the group B Streptococcus thing as well. I mean, if you look at the data, there's one or two sequence changes for so many of these viruses. It's just there's just not enough variability, right? We rely on the sequence changes to sort of help us localize these things. If they're all identical, we just don't have any information to go on, right? We just know that that's the identical thing, and that's the one that's important. And so SARS-CoV-2 right now, so we have also looked at that as, we can just run this stuff, right? And there's just not enough there's not enough, divergence yet. So we actually went back and we were hoping that there was more original SARS available, but there's just not that many sequences. Like that was 17 years ago, and it wasn't as fast that we were picking up or that we were doing the sequencing, right? So,
we need 25 of them in our institute. It was a lot of work to sequence 25 SARS genomes.
So this is exactly the question that you asked, Larry, is like, when do we get to a point where we're proactively sequencing these things so we can actually take a look beforehand? Right? Or we don't get in this situation where it's like, yeah, I mean, that's an important thing, and we just don't have any data right now yet. Like we're still waiting. Yeah, I think the cost will come down, and we are going to transition to that sort of preemptive, having predictive kind of capabilities, hopefully in the future.
I mean, PK kind of raises, shouldn't it be easier with viruses, which only have maybe 30-ish genes compared to, thousands of genes in bacteria? Is that true?
Yeah, I think it should be. So the other thing that there's been a lot of work would be in HIV as well as influenza, right? And so influenza, there's a lot of sequence available. Influenza is a very sort of interesting case because there's a very strong succession in influenza. So like they get rid of a lot of variability very, very fast in the population, right? Because from generation to generation, or from flu season to flu season, it's like just one comes out and then gives rise to everything. And then it goes back down to one and then gives rise to everything again. And it goes down to another one and it gives rise to everything again. And so I think like that also, I think we just need to learn more about how these patterns of mutation, recombination, like how fast these things are going and how much data we actually need. So I just think that Campylobacter was in a sweet spot for us, and we got lucky in that sense that the characteristics were right. But if the mutation rate was higher, if the recombination rate was lower, if the, there are all these things that we might have to adjust the analysis, and we're really blind right now as to how to do that.
Oh, but it's fantastic. You're making progress on these tools, and like you say, maybe this is a best case scenario using the Campylobacter, but what we learn, and maybe we can get more sophisticated.
Exactly.
Also, even know what to drive, what type of information you need to put it all together. I just want to relate to you, Swain, that we're getting lots of wonderful comments, commenting on how wonderful, and they really enjoyed your talk. And
okay, good. Thank you. Thanks everyone for signing on. I appreciate it.
It's been great. I think it went quite well. We didn't have any technical snafus, which is fantastic.
Yeah.
We sort of, maybe to the rest of the audience, we hopefully in the future we can actually have a more interactive, question and answer at the end. But we thought this time we would just sort of field the questions and have one moderator relay those. But we'd like to try and make this as much of a personal, interactive, seminar type system like you would have in real time, but we're not there yet, but maybe in the near future we can do that. So Reem is, and so also maybe before we sign off, I want to make sure I thank Omar Mohammed and Reem who have been doing all the work behind the scenes to make sure this thing has gone off without any technical snafus. And so thanks and tip of the cap to them. And Reem is putting up this slide to remind me to tell you, you can certainly read it, but we certainly thank you for participating in this. And we'd like some feedback. There's things we can do to improve this. And obviously this is recorded, so if you miss something or you want to relay it to your phone, by all means do so. So anyway, this was fantastic, Swain. Let me thank you once again. Fantastic talk as always. You're doing great work. Great to have a MD, PhD who can talk to the clinicians and can talk to the genomics guys. We need people with you that just have a natural babble fish in your ears, multiple languages.
Yeah, thank you for the invitation. It's just a great honor.
All right.
Thank you very much.
We're signing off, and take care, Swain.
Yep. Okay. Thank you.